So I've been working on murine coronavirus for over 30 years almost, and I really appreciate the opportunity to be part of this symposium because I really don't do COVID-19. I can't seem to forward my slides. Okay, so, so I just have two COVID-related slides, and that is to say all common circulating human viruses that really cause acute lung infections have been linked to neurological symptoms. And among these clinical manifestations are seizures, that is epilepticus, encephalopathies, and encephalitis. And evidence of virus in the cerebral spinal fluid supports that, in a few cases, neurological manifestations can be due to direct viral spread to the CNS. And early studies published by Mao et al. Actually evaluated COVID-19 patients and suggested that, about 36% develop neurological symptoms, especially those with severe disease. And the involvement can manifest in various ways: confusion, dizziness, impaired consciousness, propensity to develop strokes, loss of smell and taste, ataxia, and so on. But I just really want to emphasize that it remains totally unclear to what extent COVID-19 neurological symptoms are really due to direct CNS infection or more indirect effects, especially acute cerebrovascular disease. So the problems in providing direct evidence for SARS-CoV-2 infection is obviously the limited access to brain autopsy tissue. So at the Cleveland Clinic and both at Case Western, we do not do brain autopsies. The samples are basically limited to those individuals that suffer from severe multi-organ disease that are end-stage COVID. And the detection methods, especially in the CNS, can be challenging. Some of the antibodies can really reveal false positive staining, especially in neurons, and this really requires careful control. There's also efforts to detect virus by EM. Obviously, that's like trying to find a needle in a haystack sometimes. So in situ, viral RNA detection is most reliable. But just to emphasize again that just because you can't detect the virus, that sampling and detection thresholds may actually miss this detection. So with that, I just jump into the model that we've been using historically to just study pathogenesis of neurotropic coronaviruses, because they have been used as models for multiple sclerosis due to their propensity to mediate demyelination. So if we either intranasally inoculate or direct by IC infection inoculate virus into adult 6 to 7 week old mice, virus basically replicates to peak titers within 5 days. It's cleared to below detection thresholds. But if you look at viral antigen and especially viral RNA, it persists in the spinal cords for up to a year post-infection. And above, I'm just showing you an idea of that, the levels of viral RNA during acute infection versus persistence. There can be almost a 3-log difference. So the persistence is very, very low, so to speak, and it's not associated with detection of viral particles. The other thing is, that at the clearance of virus, the mice actually start developing demyelination, and this is an immune-mediated demyelination where T cells are really necessary to mediate this effect. And that's shown here. The mice suffer from hind limb paralysis, but ultimately they do get better again, balanced by remyelination and really sparse neuronal loss or axonal loss. So in terms of the immune response, type 1 interferon is really critical. Oh, this keeps on changing, sorry. Early on in the infection, although all coronaviruses are thought to really have a low propensity to induce type 1 interferon, T cells do the major haul here. They really are required to reduce the level below detection using gamma interferon and peripherin. And then lastly, what I'll be talking about is the essential role of the humoral response in really controlling this really low-level persisting viral infection. So historically, this is shown here, this is data published a whole while ago, but it's very clear that if you use B-cell deficient mice or mice deficient in making virus-specific antibody, that if you look at virus titers in the brain, they actually do drop, but maybe not 100%, but ultimately within a week, the virus, in terms of infectious virus, starts coming back up. Now you can give polyclonal hyperimmune serum to these mice after initial T-cell mediated control that keeps the virus under check, but nevertheless, the virus ultimately comes back up as the antibody response wanes. So that indicated that we really need probably antibody secreting cells that are locally maintained in the CNS to keep this persisting infection at bay. And additional studies also showed that only neutralizing IgG antibody was actually protective. And so we started looking early on at, are there actually antibody secreting cells in the brain? And then the top panel, you can see that the ASCs actually in the cervical lymph node and spleens on the basically peak at around day 14, but in the brain, they really don't show up until about 21 days post-infection, and they only very, very slowly increase in the bone marrow. If you look at, sorry, it's my mouse going crazy here. If you look at the cervical lymph nodes, you can appreciate that you start finding markers of germinal center formation about at the time that those antibody secreting cells are really prominent in the cervical lymph nodes. And again, that seems to coincide with those ASCs actually penetrating and going into the brain. So we had two questions. One is, how do peripheral versus CNS activation events drive B-cell accumulation in the CNS? And is peripheral germinal center really associated with these cells arriving there? So just to put everybody in the frame, because they might not be familiar with B-cell differentiation, in the draining lymph nodes, which are the deep cervical lymph nodes for the brain, the B cells recognize foreign or viral antigen. Some of them with CD4 help or without CD4 help will develop antibody secreting cells that are short-lived. Others see the germinal centers, and there, by expressing activated induced deaminase, you basically get somatic hypermutation and isotype switching, and what ultimately comes out is the memory B cells, which have high affinity but still surface antibody, and the plasmablasts, which generally tend to not circulate only for a few days and take up residency as long-lived plasma cells in the bone marrow. And those are the cells that actually provide immunity following immunization. So the other thing that's very topical is that a few chronic inflammatory diseases are associated with ectopic follicles that are sort of reminiscent of the segregation of T and B cell zones, or just provide B cell clusters in non-lymphoid tissue. And that's exemplified in rheumatoid arthritis, Sjögren's syndrome, tumors, flu, a variety of liver and lung infections. And these B cells in these clusters have diverse differentiation phenotypes, and I thought to be supported by chemokines that are generally found in lymphoid organs, namely CXCL13, CCL19, 21, and 12, and also B cell survival factors. And all these molecules can actually be found in the brain during inflammatory diseases. So our initial question is, how do these antibody secreting cells actually go to the brain? Our first option was that they just get recruited out of the germinal centers or from the lymphoid organs via two receptors that were known at that time, namely CXCR3, and CXCR4 is required for plasmablast trafficking to bone marrow, but that's after viruses have actually, or virus replication has stopped. Activated plasma cells also express CXCR3. And the other issue is there evidence for ectopic follicles? So we looked initially at CXCR3 knockout mice, and if you looked at virus-specific antibody secreting cells in the brains, you'll appreciate in the left panel here that they're very, very much lower than in wild-type mice. So it looks like CXCR3 is really required for these ASCs to traffic to the brain. If you look on the right side, in the serum, though, there's no evidence that there is impaired development of neutralizing antibodies. So the peripheral responses are fine. So the next question was, which CXCR3 ligands mediate recruitment? And we looked at both CXCL9 and 10 knockout mice. These are longitudinal sections where we stain for plasma cells using CD138. And you'll appreciate in the wild type mice here, there's really nice levels of CD138, similar in the CXCL9 knockout, but in the CXCL10 knockouts, there's actually this was like the only section we found where we found accumulation of plasma cells in a blood vessel. So further studies that were published is that CXCL10 is actually produced by astrocytes, or parenchymal stromal cells in the brain, versus CXCL9 is more localized to endothelial cells. So this could actually explain that preferential recruitment. So getting to the ectopic follicle questions, we found that if you look at the brain, to our surprise, because I used to do a lot of T-cell work, that in the beginning, there's a lot of naive looking B cells that start accumulating already actually at day 5, and they are both IgD and IgM positive, which are typical hallmarks of naive cells. And then with time, as you harvest cells and look by flow cytometry, it seems like the IgD is slowly downregulated. We have single IgM positive cells, and ultimately these IgD positive cells, it's driving me nuts, sorry, that accumulate. So this could be because you can get local differentiation in the CNS. But when we looked at these early B cells and also at later time points, it was very clear that at day 7, these B cells tend to be more at perivascular and meningeal sites. You see some in the middle panel where the arrows show that the B cells start going into the parenchyma, and by day 21, actually the vast majority are in the parenchyma. When we looked at the distribution and tried to quantify this, if you look at the IgM panels, the left one shows IgG staining. Again, most of those B cells are in the parenchyma. And if you look at the distribution of both IgM or IgG cells in the CNS between perivascular versus parenchymal, you'll appreciate that the IgM or positive cells stay basically localized in the meninges, versus the IgG ones actually do go parenchymal. And that happens more at those later time points where we know we get more differentiated cells in the brain and in the spinal cords. We also tried to look at markers of follicle formation. Just to say up front, we could not find that. We found rare clusters of B cells, and there were proliferating cells, but they were restricted to non-B cells. There's like the very rare B220 positive cells that is apoptotic positive. But in general, it's the CD3 cells that undergo proliferation. We also found very little evidence for caspase-3 in the lower panel, indicating apoptotic B cells, which would be the case if B cells turn over rapidly, like in germinal centers. And we also found no evidence for follicular dendritic cells or expression of the germinal center marker GL7. So, but what we did see is that we looked at B220 and T cell staining using CD3, And it became clear that some B cells are actually seem to interact with CD3 cells. And in panel C, you can see that nicely, looking at more the horizontal view, both to the right and at the bottom panel of C, where you can see that there's some overlap in the colors indicating membrane interactions. So we think that even without ectopic follicle centers, that CD4 T cells can maybe give extra signals to these B cells to then actually go parenchymal. So is germinal center formation in the periphery the major driving force for B cell subset accumulation in the CNS? And to address this, we actually looked at reporter mice for activation-induced cytidine deaminase. I already told you this is an enzyme necessary for somatic hypermutation and class switch recombinations during germinal center reactions. And we used mice in which the AICDA reporter actually expresses inducible Cre recombinase, and they're basically bred to Rosa DD tomato mice. So if AID is expressed or the enzyme is expressed, the promoter is turned on, all those B cells will ultimately fluoresce with a red color. So this is a really nice surrogate to really look at antigen primed B cells in lieu of having to do memory B cell responses, which is difficult for MHV since the major receptor is highly expressed on B cells. So using this model, we can administer with tamoxifen either at the onset of infection to cover all the AID expressing cells throughout infection, or do a timed tamoxifen injection after infection. So when we look at tamoxifen treated infected mice throughout the whole infection and give tamoxifen every second day, you'll appreciate the numbers of B cells that are DD tomato positive and draining lymph nodes sort of increase at day 7, more so by day 14. This is shown in the left panel, and they make up about 7%. And this is sort of typical for any infection. If you look in the brain, you'll appreciate that the numbers of sort of AID positive or DD tomato B cells really increase at day 14, and even more so after the onset of germinal center formation. And in the spinal cord, it's very clear that the vast majority of B cells that actually show up during this persisting phase have undergone hypermutation to some extent and isotype switching. So that would really indicate that there's very sparse bystander recruitment of pre-existing memory B cells or ASCs. So the recruitment of these cells is really specific to virus-specific, or at least B cells that have recently undergone germinal center reactions. And to just show the distribution of memory B cells versus ASCs, looking at ASCs using the CD138 marker, and for memory B cells, they just don't have the IgD and are CD138 negative in blue. It became apparent that in the cervical lymph nodes, actually we have AID positive cells at 7, but they really never show up in the brain. By day 14, again, when we see nice germinal center formation, the vast majority of these are memory B cells in the cervical lymph nodes. Also, what shows up in the brain and spinal cord, this is the red box, are mainly memory B cells. And then with time, although the fraction sort of always favors memory B cells in the cervical lymph nodes, what you see in the brain and spinal cord is really predominant recruitment or retention of antibody secreting cells. And I just want to say that we find no evidence of AID expression in sorted B cells from the brain. So we really think these are being actively recruited from the draining lymph nodes. And finally, we also did some time tamoxifen treatments going late up to, so we don't treat mice with tamoxifen until day 35 till 42, which is more recent data. And you'll appreciate if you look in the lymph nodes at all these time tamoxifen treatments, even late out, we still get nice germinal centers. We think these cells are all virus specific. And importantly, when we look at the brain, you'll really appreciate that these AID positive cells even show up late. So there's continuous recruitment of these cells into both the meningeal perivascular sites as well as the parenchyma. And a lot of them, as the arrow shows, tend to be B20 negative. So they probably are real antibody secreting cells that are more differentiated. So just to finish off the talk, what we hope we showed or tried to convince you of, that the brain is really a secondary site for B cells to actually be retained for a long time. The B cell turnover goes from early naive looking cells to retention of memory B cells and plasma cells. So what we think is happening is that early on, just a little bit of activation drives these what look like more naive or early activated B cells to be recruited. We think those are chemokines that recruit these that come from the stromal cells, namely CCL19 and 21. We're just working on that to make sure they light up in the meninges. At least we see them by PCR, but they don't really go parenchymal. And then as germinal center formation sort of starts in the draining lymph node, that only these cells actually ultimately are also recruited to the CNS, that different chemokines, for example, for the antibody secreting cells, CXCL10 is involved, and that these cells are the major ones that really tend to stay in the brain or the spinal cord as long as virus persists. And we're sort of still looking at what is the role of these non-antibody secreting memory B cells. Can they spontaneously turn into ASCs? And we know that they are really required to maintain even very, very low levels of infectious virus in the persisting state or in the persisting state. So again, we can never find infectious virus, even if you irradiate the mice, only if you take away these B cells. So with that, I'd just like to thank the team. Many have moved on. Most of the studies were done by Jeff Atkinson and Krista DiSano in the lab. They've moved on to do postdocs. Agnes Renaud gave us the beautiful AID transgenic mice for this project. And below is my wellness exercise team. And with that, I'll take any questions. And I apologize for my mouse being a bit sensitive and moving slides forward.
Thank you, Cornelia. This was a wonderful presentation. Looking forward to questions.
Andrea, there's a question.
The Q&A.
The Q&A.
So how do you house your MHV infected mice at your institution?
So they are, we have a non-barrier facility where we breed most of the mice. We sort of see if the mice don't get sick or have no virus, they probably have pre-existing MHV, which is very, very rare. We've actually not seen that since being at the clinic. So they are just in a normal animal facility, not barrier. I hope that answers the question.
I guess it. So, microglia in CNS, is there a difference in the way we operate, in terms of antigen present? Any other differences?
So I haven't looked at the B cells, whether they can really present viral antigen. What we have done is take out the microglia by flow cytometry from infected brains, and also looked at sort of the CD45 high CD11b myeloid cells, which are a mixture of dendritic cells and infiltrating monocytes turned macrophages. And we have CD4 transgenic T cells for one of the epitopes. And surprisingly, we cannot get microglia, at least taken directly ex vivo, to activate either the virus-specific T cells or myelin-specific T cells, but the infiltrating cells and the dendritic cells do.
Right.
So we don't know whether those memory B cells might sort of function as additional antigen presenting cells. They're not there at very high numbers, so we need a lot of mice to sort those out and do the similar experiments. But that could be a possibility of their function. We just don't know. Clearly, they're very important. Also, in COVID-19, people are looking not only at plasma cells because they tend to have a more limited repertoire than the memory B cells.